So our next speaker is Jens Krause. He is professor of fish ecology at Humboldt University and also the department head of biology and ecology of fishes at the Leibniz Institute of Freshwater Ecology and Inland Fisheries. His research is focused on mechanisms and functions of group living in animals, which he studies in the field, in the lab, and also using models. Thanks, Jens.
Yeah, first of all, I would like to thank the organizers for the invitation. It's a great pleasure to be here. So I want to talk a little about some of the work that we do on fish and humans, and we keep alternating between the two systems because we often find that experiments on fish will inspire experiments on humans and vice versa, and I hope to show that in this talk, how that works. And by way of introduction, I want to show you a little experiment that Ashley Ward and co-workers and I did some years ago, which brings me to the actual stuff that I want to show you today. So this was a simple experiment on mosquito fish, tiny freshwater fish, a few centimeters big. We looked at their decisions in groups, and we had here different group sizes, starting with a single fish up to 16. They get introduced into this maze and they make a right-left decision. And in one arm of the maze, you have a cryptic predator that they need to detect and avoid. And when they do this as individual fish, they're not particularly good at it. Very often they overlook the predator. You can see here individual fish make fairly poor choices. However, individual fish in larger groups get it right almost all of the time. This is not particularly new. I mean, there were already studies from the 1970s showing stuff like this from bird flocks, that they can detect predators earlier and initiate evasion behavior earlier on. But the interesting part came when we looked at the time that the animals take to make these decisions, because surprisingly, in larger groups, they take considerably less time than individuals. And I think all of you go to staff meetings regularly, and you all know that normally collective decisions are a function of group size. So the fish have found apparently a different way of dealing with this problem. And one of the potential answers here is single individuals, when they move down this channel, they hesitate a lot, they are multitasking, they're looking for a lot of things, obstacle avoidance, and they look out for the predator, maybe they also look for food and so on. However, when they're in groups, they can actually only see a tiny slice of their environment, and for this space, they can make very accurate decisions and copy them from each other. Okay, when we submitted this for publication, the reviewers liked this, but one person said, well, I mean, these fish avoid the predator when it is there, that's this curve here, but how often do they avoid a predator when it is not present? And I thought, what a pain. I mean, why should I think about how my fish avoid a predator that is not present? Well, we got this published, I forgot about the comment, but later on I returned to it and I thought, actually, this is a really interesting question. And so if you look at an animal making a decision under predation risk, so take these fish, a shadow goes over the water. This could be a butterfly that is harmless. The fish should stay and maybe continue foraging. But if this shadow indicates a kingfisher, a predator, they should disappear and take refuge. Now, how do they know which is which? If an individual fish becomes very sensitive to all shadows, it will waste a lot of time running because it's going to avoid a lot of butterflies, a lot of harmless stimuli. But it will also avoid all potential predators. If it hardly ever responds to the shadows, well, I mean, it will never commit the false positive, avoiding a harmless thing, but it will frequently be under attack by real predators. And when we thought about this, we realized this is actually a problem that humans frequently have as well. If you take the case of a doctor, for instance, you go to the doctor, you think you might have skin cancer, you have a mole on your skin, the doctor will take a look at this, they see the dark part of your skin, they need to make a decision. Do I really initiate treatment in this person? If the person has skin cancer, you initiate the treatment, it's a true positive, that's what you want. But if they initiate treatment despite the fact that you don't actually have the condition, well, that's clearly undesirable. But it's very difficult for a doctor to do both. Always initiate treatment when it's really warranted and never when it's not. And a lot of security decisions are also based on this, fall into this category. At airports, we all know it. They often look into our bags despite the fact that we don't have anything in there. But they also need to categorize as in terms of are we a security risk or not? So we constructed an experiment with humans where they need to evaluate a situation. One is harmless. They need to detect here these spiny fish on a slide of many individuals. If the situation is harmless, they need to press a button on a gadget and tell us they want to stay. If it's a dangerous situation, if this seven-spined individual here is present, they should press leave. We show them many of these pictures, and what we do is here we only show the whole group the number of individuals that want to leave. Then they have a moment to reflect on that, and then they all vote again, and we look at the collective choice of people given this social information that they got about how many want to leave and have assessed the situation as dangerous. If we look at choices in these contexts, one example, for instance, here, 60% of the time people get it right. That's our true positive rate. They leave when the situation is dangerous and when they should leave. But in 30% of the time, they wanted to leave when it was a harmless context. So the question is, how can they deal with this? How can they make better choices? And it turns out if you put a quorum in between the true and the false positive, so you only leave if at least Q% of the group want to leave, and otherwise you stay, then you should be able to increase the true positives to a very high level and bring down the false positives to a very low level as a function of group size. Now the question is whether people are really capable of spontaneously doing this without knowing the theory. And so we did this experiment, and we found, yes, in fact, if you average over many runs in this experiment, the black bars here indicate the individual response of all members without feedback. And if they then vote with feedback, they can dramatically increase the true positive, and they are capable in most cases, we have a few exceptions here, but in most cases, they can dramatically reduce the false positive for these situations. So that's something a single individual cannot do. And what's interesting about these cases here is the true positive rates are actually very low. They're below 50%. So you could never amplify up using a Condorcet theorem where the expectation is that each individual has at least a 50 or better % chance of getting it right, because the individuals here are below or around 30% with their individual assessment. So this, in a way, is a collective decision that can work if just a minority of individuals knows how to assess this correctly. So in this situation, for the humans that I showed you, for the individuals, it's good to solve the problem collectively. It's good to solve the problem. But what about competitive situations? And I want to show you one where we return to our fish. We work a lot in the rainforest in Trinidad. You see a typical stream here. We have these fish, guppies, and the males have this orange coloration. That's what makes them attractive to the females. But this is a pigment they cannot synthesize. They need to ingest it via their food. And this makes food very popular that is red with these fish. And we can now present them with red stimuli that really contain carotenoids and false ones that don't. So we take tiny red pieces here. And I just show you the choice that they make when they see these two red objects in their environment. And we tested again different group sizes. And this situation is a competitive one because while you are trying out one stimulus, the others might have already eaten the valuable one. So this is what this looks like. And the fish respond very strongly to these stimuli. So we can see here that a single individual or small groups are quite poor at discriminating true and false positive. But as the groups get larger, the fish become increasingly better to make these choices. We observed them in this case for 2 minutes, so we made the stimuli fairly large so they don't consume the true stimulus completely. So it might also be interesting to see what happens if we give them less time, because the true stimulus actually gets consumed in that time. So I want to show you a bit of an outlook where we go from here. With the human experiments, we've become very interested in actually testing this collective decision-making algorithm on medical data sets, looking at choices of doctors that they make when they assess cancer. And this research I'm doing with Max Wolf and Ralf Körbers, and they've produced some interesting results on this already. This algorithm seems to work for real-life cases, not just for finding spined fish on a slide, but I want to leave that to them to show that in future. I want to talk a bit more about what we do with a fish and how we investigate collective decisions with these fish. And we thought a great way of doing this would be to have complete control over some individuals in the group. And we can do this by developing an interactive robot. So this is a little fish here, a few centimeters, and real fish accept this as a conspecific. So we can enter this into a real-life fish school, and I want to show you how they interact. So here's our robot. So this is a top-down view of a tank, 1 meter by 1 meter, 10 fish, and then it takes control. And it actually takes them all back into the box. Unfortunately, only works in the lab. Can't perform these magic tricks in the ocean yet. Okay, but what about the leader making a maladaptive choice? So now it's leading to food and then tries to take them away from the food, okay? Something that's not so good. You see one fish actually hesitates and then does the right thing, so they ignore the leader, okay? And this is also a function of group size, interestingly. Okay, but what's new is we can make this thing truly interactive in these situations that you've seen so far. We only programmed the robot to swim a certain pathway, but it cannot actually respond to what the live fish are doing. And now we can track them in real time, and the robot can respond to what they do, which means we can actually use the robot to embody collective behavior strategies that are published in the literature. So this is, by the way, a different species here of fish. These are guppies. And we gave the robot here the instruction to stay in the center of the group, but we could program now any mixture of leader and followership. And one of the first things we want to investigate here is, for instance, what is the optimal leadership strategy as a function of group size? How much do you need to lead and how much do you need to follow to move groups of different sizes from A to B? I should mention here that we are doing this work at the moment with Tim Landgraf from the Free University, and as part of the new Humboldt-Princeton Exchange Initiative, I hope to do some work with Naomi Leonard on this and exchange members between our labs. And then the other system I want to mention is one that my colleague, Robert Arlinghaus, is pushing. Our institute has a lake where we put listening stations inside. We can put hydroacoustic tags on individual fish, and we can track them in 3D. Every few seconds, we get a 3-dimensional position for every fish, and we can track them for up to a year and look at their dynamic social networks. We can look at how their behavior is influenced by their personality types. And because we can do it over extended time periods, we also hope to look at the fitness implications of collective choices that these animals make in this lake system here. The tags are quite expensive. They cost a few $100s each. So there are limits to how many individuals we can tag. But it works surprisingly well. And the last thing I want to show you is something I'm very excited about at the moment, because we got these new videos. We are looking at predator-prey interactions in the open ocean. We look at how prey collectively defends itself against predators like this 2-meter sailfish here, and the strategy that the predator then uses to overcome the defense of the group. And they use the strategy of battering these fish. So this is a group of sardines, and the sailfish puts the bill inside. You can see this here repeatedly, and they really hit these fish very hard. You can see the scales and the tissue flying here. And this is played out over hours and hours. And the interesting thing is here, we measured this, and we found that the sailfish will hit individual sardines in the school up to 40 times before they actually get consumed. And so this really means death by 1,000 cuts, because they really strip these sardines down and skin them alive. And so we are looking at these interactions, what the fish do to avoid this fate. And finally, I would like to thank all my collaborators, the empiricists and theoreticians, and those organizations who provided the funding. Thank you very much for your attention.